This is a production of Cornell University.
I feel very fortunate to be here this time of year. Ithaca is beautiful, and it's my pleasure to talk about palms. I titled this talk Integrative Studies in Palms. I don't just do field research in palms, but it is my group of focus, and I hope after this talk you'll feel motivated to at least explore some aspects of this fascinating group. I use palms to navigate the timescales. Timescales, I would say my base is in population phylogenomics and population ecology, but I work with geologists in projects about geogenomics, historical demography, biogeography, and with chemical ecologists and entomologists on the reproductive ecology of palms. And I try to make all of this information available for conservation plans for endangered species. So I'm going to introduce the palm family, at least the relevant aspects of the family, and then talk about 3 of the topics I've addressed, and even some of the current work I'm doing. Not all of it is finished. And talk a little bit about the future and leave some time for questions. So this family, it's a well-known, pretty famous family, I'd say. So most of you, I'm sure, know it's a mainly tropical family. Some species are subtropical, but diversity isn't evenly distributed through the world. So there's 2 diversity centers, Northwestern South America. So here and then Southeast Asia. It includes 2,600 species and 188 genera. And it has evolved over 100 million years. And in general, palms are adapted to warm environments, so high mean annual temperatures, low temperature seasonality, and the coldest month has to be above 5 Celsius because they can't really cope with frost. Some very interesting aspect about palms is that they can be very abundant. So in Amazonian forest, where this study was done, taking data from all of these plots in Amazonia, they list the 20 most abundant, frequent palms are not trees, but tree-like trees in the Amazonian basin, and 7 of the 20 are palms. And the most abundant tree-like stem is Euterpe precatoria, which is the acai palm. So they're structurally very important for tropical ecosystems. However, we found that in many cases, these extreme abundance that leads to monodominance is related to disturbance, to human disturbance in many cases. This has led to the hypothesis of Amazonia as planted forest, where these forests are enriched with species by human management of these forests. This was certainly the case for Ceroxylon, where we worked in these central cordillera in the Andes and in other spots along the tropical Andes. Even if they're very abundant, very frequent, there's still discoveries being made, so new species being described. This is one of last year's gems. It looks like a very ordinary palm when you look at it, very ordinary understory palm, but because of local people living around these forests, that led to the discovery that, well, actually the fruits are foraged on by wild boars, and the flowers are underground. We don't know who pollinates these palms, and this just highlights the importance of working with people around biodiversity because they know a lot more about the natural history of species around them. Perhaps because they're so abundant and structurally important, peoples from the tropics use them in many ways. So to build their homes, this is the inside of a home, and you can see the floors, the walls, made of the Rhyartia deltoidi, the roofs, the entire homes, fences. These are just some examples. Elevated gardens, Ramoetinia calbrieri, a beautiful palm, stairs to climb up to those gardens, and even what's used inside homes. So this is a part of a broom being made, so brooms, and even what's eaten inside of those homes. So this is the peach palm, a wonderful source of many nutrients for many communities in the American tropics. So it is of no surprise that palms are named in just every culture, in just every language. These are non-Spanish names for the peach palm in the Colombian Amazonia. So you can see you'll find some similarities between these words because some of these languages are related, but because they're so important and so used, so fundamental for human livelihoods, they have a name. They're named. So this is an example of a tropical home. This is very common, and palms are everywhere in the tropical home. However, we're losing forests, and without forests, we lose this backup resource for rural communities, and also this incredible opportunity to learn from palms. So this is even more concerning given their conservation state. So in this study, 80% of the world's palms were studied, and 56% of them were found were in some endangerment category, regardless of the method used to classify them. So the red triangles and then the bars, they're showing different methods. You always have a high percentage of the threatened species, and they fall in many categories, whether it's evolutionary distinct species, functionally distinct species that are not replaceable, or they're used by humans for culture, food, utensils, medicine. So regardless of their category, you always find some large percentage of the species endangered. And this is worse for the regions having over 10 species. So the species-rich areas in the world have higher endangerment. And this is the areas where people rely on them most. So my research addresses the general overarching question of how do species and populations persist in time and what they can teach us about their changing environments. This would be like the overarching question. And of course, with a focus on palms. So I'll talk a little bit about my research now. So I've always been intrigued by elevational gradients. If you've worked in the tropics, you can perceive the high turnover from elevation gradients. They're absolutely interesting. They're very variable between different slopes and different mountains. And particularly more so because elevation gradients offer climatic gradients that go along with them. And because palms are so well adapted to warmer conditions, it's very striking that some groups are adapted to cool environments from the highlands. And this particular genus, Hyphanes, it includes 35 species, and they live from 0 meters above sea level in mangroves to 3,200 meters above sea level, close to the tree line. So we wanted to understand how this adaptation to cool habitats evolves in their phylogeny. So we built different phylogenies with different data sets. We had Sanger sequences, but we also put together this phylogeny based on hybrid capture using 4,000 nuclear coding and non-coding regions. This is the summarized species tree, astral tree, and I've colored the clades. And what this shows you is that all clades, so each clade is a color, and what you can see is all clades have evolved to live or to cover the elevation gradient. So every single clade repeatedly has a species that lives in warmer conditions, mid elevations, and high elevations. And this happens repeatedly for every clade. And indeed, when you look at temperature-related variables, here indicated by principal components, or temperature or precipitation-related variables, you see the precipitation-related variables have higher phylogenetic signal, whereas temperature-related variables do not because they flip and flop around branches. So this is the model that explains how these are evolving. So the different clades basically have species covering the entire gradient, and adaptation to different temperatures can evolve in all of these clades. However, this all happens by conserved low precipitation seasonality. So these palms can evolve if there's still low precipitation seasonality. And this indicates a little bit about what would be a big threat to their future, because they can't really cope with higher seasonality, whereas they seem to do well in many different temperatures. So this has allowed complexes like this Hyphanes hirsuta that we've been studying and that we're continuing to sample to cover the elevation gradient. You have populations at the tree line and populations by the sea. And we want to understand what the differences in populations at different environments can tell us about adaptation to different climates. Another group I've studied for years, Chelsea mentioned it, is Ceroxylon, the wax palms. They're very fascinating because they live in extreme environments for palms. They live in high elevation forests, cool forests that are constantly covered by mist and clouds. So this is extreme for palms. They're also dioecious, so they have female and male flowers, and they're only slightly dimorphic. So you'll see this is the female flowers and the male flowers. They're very similar, but the male flowers are just slightly drooping, and they can reach to 3,500 meters above sea level, which is 1,500 feet, so very high in the mountains for palms at least. We wanted to understand how are these populations making it? How are they persisting in such an extreme environments? And more so because we sometimes find congenerics coexisting within the same forest. So you even find syntypy in these extreme groups. So we followed a forest that had 3 different species from Ceroxylon living together. So Ceroxylon parvifrons in the middle, and Ventricosum in the left, and Vogelianum. And you find viable populations of all 3 living together at 3,200 meters above sea level. We followed their flower visitors and their phenology for 3 consecutive years, every month. And we found that 50 morphospecies, 50 insects, visit all of the 3 species. We found no differences between the 3 of them. But the effective pollination services are mainly carried out by Mistrops rotundula, which is this small miniature beetle. And this is how it goes. So the male inflorescence emits these alkanes, these aliphatic compounds, and they attract the beetles. They come by swarms, they feed on the pollen, then they mate in the inflorescence, but then they're attracted to the female inflorescences by the same scent, but then it's by deceit, right? Because they arrive at the female inflorescences and there's nothing to feed on there, but then they pollinate the flowers. And this beetle is important in the pollination of all 3 species, which are closely related. So we wanted to see how they partitioned this resource. And it's temporal partition, which is classical. So we have the 3 species in 3 colors, and this is the summary of the 3 years. And you can see they have different flowering peaks for male inflorescences, female inflorescences, and fruits. And flowering peaks occur outside of the rainy season. So here you see the rainy season. It's these yellow peaks. That has probably to do with volatile emission being more efficient during the drier seasons. But we did find there's some overlap between species. So there's the potential of pollen from species 1 to end up in the stigmas of species 2. And because they're closely related, we had some questions about this. But this system is highly efficient. So reproduction is phenomenal in the sense that the smaller species can produce 3,500 fruits per season, and the larger species, Ventricosum, 10,500 fruits. And these fruits feed toucans, toucanettes, green jays, parrots. And this even happens in areas that have been deforested. So we've also worked in this other site, the Tochecito Valley, which has been disturbed, as you can see. And every season, during masting season and non-masting seasons, there's efficient reproduction. So we think these 3 species, at least, are a fundamental habitat for the beetles. So we had some questions after having done this work. These 3 species are taxonomically very easy to identify, even from far. So this is Vogelianum and this is Ventricosum. And with very little training, you can tell them apart even from the distance. So they're taxonomically good species. And they're different. They're the same in every single population where you visit them. So if you collect Ventricosum in Ecuador or in Colombia, they're exactly the same. In parvifrons in Bolivia or Colombia, they're clear species and taxonomically, but are they really genetically isolated because they have these overlapping flowering peaks and they share the main pollinator? And if there is gene sharing, is it adaptive? So we sampled populations all throughout their range. So these populations are patchy, you have some scattered individuals. We wanted to at least sample all of the population patches. We sampled all but one species that was more recently described, with emphasis on the highland species complex, which is a monophyletic complex, and they sometimes live in symmetry. And we use this same genomic data set. So these are the highland species. You have Vogelianum in green and Parvifrons, and you see it's highly overlapped. And then you have this Quindiwense ventricosum seriferum. They kind of replace each other latitudinally, and they all live at the same elevation between 2,000 and 3,500 meters above sea level. And this is the underlying cartoon phylogeny for this species. And what we found is that we have pure individuals for Parvifrons and pure individuals for Quindiwense, but then Seriferum and ventricosum in dark blue here, they fall everywhere in between. This hybrid index is admixture-based. So this led to the idea that we have really 3 species and then 2 introgressed forms that we've been calling in taxonomy ventricosum and seriferum, but they're actually just admixed forms of the other 3 species. When you bring genetic data for all 3 species, Vogelianum, Quindiwense, and Parvifrons, shown by these triangular graphs, you find some pure populations in the corners, but then you find, indeed, ventricosum and seriferum right in the middle. So they basically have shared, they're sharing genes with all 3 species, which we could understand through what we knew from their reproductive ecology. Because admixture-based methods aren't enough to detect hybridization or introgressive hybridization, we also use the statistics, or ABA-BABA statistics, which is the proportion of derived versus ancestral allele sites for ABA and BABA patterns. And what we find, and then the F4 ratio, which is an excess in allele sharing, so an excess of D. And these larger circles are basically showing you where there's more introgression, and we find that's particularly so when we have Parvifrons and Seriferum, which was our initial observation from the field. And when you map those populations to their geographical locations, we find that the pure populations are at the center of their distribution, whereas the most admixed populations are at the periphery of their entire distribution. So you have the entire distribution, and towards the margins is where you find the more introgressed populations. We wanted to see if these introgressed regions coincide or matched with sites under selection by these evolutionary models, by Liu and collaborators, and we found that's rarely the case. So introgressed regions are normally not under positive selection. And the way we understood this is, well, we have 3 species that can share genes, right? But there are differences in their population sizes. So we have two species with large populations, and then 1 species with a very small population. The species with the smaller population size will amplify the introgressed regions because of drift and demographic effects. So this is called the genomic neutralist hypothesis, and it just highlights the essential role of inefficient reproductive barriers. They're sharing genes. Why? Because they don't have efficient reproductive barriers in place. And this is amplified through demographic processes because you have smaller populations in the periphery. So as you move out of the center of their distribution, populations become smaller and smaller, and individuals become sparser and sparser, and thus these introgressed regions can become more amplified in the receiving population. So this was interesting because it also shows that for some of these species, a way of maintaining diversity levels is through gene sharing, specifically so in areas where populations are smaller and individuals are more sparser. So it highlights also the importance of having congenerics in the same area. It's not just about protecting one wax palm, but all of the wax palms that live together in that forest, because they can potentially share genes. Okay, now I'm going to talk about geogenomics. This is integrative work I do with geologists. I really enjoy doing integrative work and working with geologists because I learn a lot from them, and I'm always amazed by the level of complexity of their work as well. And doing field work with them just shows you a whole different things that you wouldn't have looked at. So it's been really good to work with them. And with them, we've been working on the notion that the Andes, although they seem like a continuous 7,000 kilometer mountain chain now, they haven't always been continuous in the past. So they have two different models for Andean evolution. One is just a continuous chain that gradually uplifts. And then there's a second model where you have isolated mountains that form and then later connect. And these two scenarios offer different evolutionary scenarios for biota, right? If you have a continuous mountain chain, then you can have dispersal occurring throughout time. Whereas if you have uplift and then connection, then dispersal of species should be episodic. It should happen at a given time frame when the mountains connect. And bird data, there's a lot of bird data and on the distributions of birds. So bird coordinates, bird occurrences have been used to create these bioregions along the Andes, for birds at least. And you can see there's a lot of turnover. If you work in the Andes, one thing you will perceive is that there's a lot of turnover. And this is what this map is showing from bird occurrence data. Within short distances, you have turnover of species and even phylogenetic breaks. So we wanted to study these phylogenetic phylogenetic breaks, and we wanted to study that with geologists using a group that is widely distributed and across the different breaks. So we use genome data, which includes different names. It's more of like a complex. It's a complex because it's very morphologically variable. So you have the very small adults. That's an adult. You can't really see here, but that's the inflorescence is here. So this is a meter tall adult, but you also have taller adults, and the pinnae, the leaf segments, can look many different ways. So we've described this whole complex as a hypercryptic radiation. It includes many genetic groups and very complicated morphology, but they do grow across these breaks. So we sampled them in all of the populations we found through the Andes of Colombia, from the Sierra Nevada de Santa Marta here and into the Eastern Cordillera, Central and Western Cordillera, and what's called the Colombian Massif. And we found there's 4 genetic groups. And these 4 genetic groups, when working on their distribution with geologists, we found these genetic breaks coincide with the main faults of that occur within these areas. And why are faults interesting to us? Because if mountains were segmented, if mountains were formed by different mountain segments that only later reconnect, they would have been broken or segmented along these fault lines. Because topography doesn't grow in the same way along faults than non-faulted areas. So you would have differences in relief just along these lines. And when you look at those genetic groups, basically they occupy the same climatic space in terms of temperature and precipitation. You can see it on both of these PCAs, but also here. All of these genetic groups basically occupy the same climatic space. But if you take morphological traits and you map all of the genetic groups together, they overlap. But if you take the sympatric groups, there does seem to be some morphological displacement. So if groups are sympatric, they have displaced morphologies. If they're allopatric, then they overlap in terms of morphology. We wanted to see if these sites under these FST outliers for higher divergence, Tajima's D outliers, which were showing us drift and population reductions, and sites under positive selection, if they mapped to the same sites in the genome. So these are the chromosomes. And what we find is that they don't. So they rarely overlap. So few sites are under divergence and selection. Except 2 genes that play a role in flowering times. So again, no surprise, groups are flowering times are playing an important role, and then response to environment, so chill and light responses. So what's the role of faults then, right? Because we had been working with geologists. So what we think is these faults create initial vicariance, so initial allopatry, but then there's these divergence between groups is reinforced by drift and by, in parallel to morphological displacement and phenological displacement. All right, so we had this system where we were thinking about ambient segmentation. The geologists were sampling, and we were trying to understand if they had been continuous or not, and what plants were showing us, right? Because we want plants to inform us about their changing environments. And we had the bird maps, we had our genetic groups for Geonoma, and this wonderful paper came out. It's a paleogeographical reconstruction of that particular area where we had found group turnover, genetic group turnover. And in this particular area, there's what was called the Trans-Andean portal, which connected the Amazonian basin with the Pacific. So this might seem like a small thing, but it's actually huge. It's a greatest rainforest connected to the Pacific, which means the two of the most plant diverse regions connecting through this low pass area where the Andes weren't high enough to isolate species. And this occurred until 6 million years ago. So it was late Miocene, which would have affected the distribution of many species. So we used our data set from Ceroxylon. We had sampling set from all of the Andes and across the Trans-Andean portal. We built several phylogenies again, and we used biogeobears for biogeographical reconstructions. And indeed, we did find that wax palms were in the Andes since the Miocene. So they were already there since the late Miocene, but they were there south of Ecuador, so in Bolivia, Peru, and Ecuador. And it was only in the Pliocene that they could disperse into the Eastern Cordillera of Colombia, and only between 3 and 2 million years that they could disperse into the Central and Western Cordillera. So it was episodic and it was late. So dispersal was not continuous throughout their history. All of the highland clades repeatedly dispersed to this area at the same time, except this lowland group, which is not indicative of highland continuity, right? So we went out with geologists and, well, you can't really see too much here. I see it there, but this is a map of that area in southern Colombia where the Trans-Andean portal was. And we sampled all of this area with, so it's like a 3-degree longitude and latitude area. And we sampled with geologists and we found these ignimbrites, which are rocks that are ejected by large volcanic eruptions, caldera-forming volcanoes. And they're thrown, and you can date when they made surface through geochronology, which is basically uranium to lead decay in a mineral called zircon. And with this, you can know the age of the rocks when this volcanic eruption occurred. And interestingly, all of these rocks were ejected in a very large perimeter, in a very large area, which I'm showing here by the degrees, between 2 and 3 million years. So it's exactly at the same age. And not only did we find evidence for this large-scale volcanic eruption, we also found that these volcanoes are capable of producing lava flows that can fill up low valleys to 1 kilometer deep. So if you have a low pass, but then you have a large-scale magmatic addition, which can fill up the low valley, then you can connect mountain segments. This is added to bulging and other processes that occur along with volcanism. So our paleogeographical model is basically, you did have highlands before 5 million years. You did have mountains, but they weren't connected. And through volcanism, across these centers, you would have magmatic additions that basically connected segments and allowed species to disperse in this particular time window. So because we had worked with faulting and volcanoes, we teamed up with bird researchers, with bird researchers who were working on this hybrid zone in the same volcanic area, volcanic center. And we wrote this perspective, which basically ties geological processes to life evolution processes. So cortex bulging, valley filling, lava flows, tephra falls, gas and aerosol emissions, to diversification, extinction, and dispersal across different time scales. So this is just to bring together processes in the geosciences and in the life sciences, which is what geogenomics does. Now switching gears to the Amazon. So this is the Amazon, but right close to the Andes, so the Amazonian Andean foothills. And we're particularly going now to the Vaupes Arc and to the southern expression of the Vaupes Arc, which is this Chiribiquete range that I'm showing here. So it's not 5,000 meters elevation, which we had for the Andes. It's a smaller mountain range, but it's enough to have rivers flow north and south of it. So it does affect drainage evolution and sediment the transport of sediments, etc. So we're in this corner again of northwestern South America, and these are the Andes. And you have this perpendicular series of mountains that are called the Vaupes Arc. And this is the southern expression of that arc, which is called the Chiribiquete range, is what you see here. This whole range uplifted at 8 million years ago. And some of these rocks that are exposed are older, but the arc is thought to be 8 million years old. And this creates the water divide between Orinoquia and Amazonia, which are two very important biogeographical regions for South America. So Orinoquia is seasonally dry savannas, while Amazonia is closed canopy rainforest. So they're extremely different, just because of this low relief created by this arc. And a parenthesis here is that this Chiribiquete range hosts one of the most extraordinary archaeological sites, kilometers and kilometers of rock paintings that have been dated at 20,000 years ago to present. And in these rock paintings, you can even find animals that are now extinct, like the giant sloth. So they're really old rock paintings. This is one of our sampling sites. And for Amazonia, there have been different factors that have been studied that influence the distribution of its biota. So current climatic variables, vegetation structure, which doesn't lead to much turnover. And one of them is river and barriers. So the effect that river basin evolution has on structuring composition. And we wanted to explore that in particular, study that in particular, because what was found with these maps for amphibians is that this is the water divide. So what you have here in dark green and in yellow, that's the water divide between Orinoquia and Amazonia. And you can see the compositions are very distant. They're adjacent regions, but with very different compositions for amphibians. So we used this group, Astrocarium. They're spiny palms. Some of them are very abundant. Very important in Amazonia too for local economies. This is again the water divide. What you have here is again that water divide along the Vaupes Arc. So we've been sampling Astrocarium ginacanthum, which is an understory palm that you see here, Chambira, a tierra firme canopy palm, and Jawari, which is a riparian, lives along rivers, palm of the same genus. And we've been sampling along the rivers of this whole region of the water divide. We've done 7 rivers so far, covering the water divide. And I just wanted to take a minute to show you Astrocarium jawari. It always lives along rivers. You can't really sample it by walking. You have to sample it from the boat because there's nowhere to stand on. And this is how complex vegetation is there at that site. This is Astrocarium jawari, showing a little bit of its crown. And what we found is, even if this relief system isn't as high as the Andes, it does structure populations north and south of the relief system. So in all of the 3 species, even if they have different habitats, if they have different life histories, the individuals do group north and south of the divide. So this is that relief system where it would be. And then you have the southern populations and northern populations, north of and south of the divide. South of the divide, north of the divide. In the river species, it's more complicated. You have like a softer break between north and south, and we think this is because of its habit. So we do have like two groups, but the cut is softer, and we think it has to do with the fact that fruits land on water, and then they remain viable for many months, and they can float on rivers, and then dispersal is more efficient in this species. FSTs are low between north and south for all of the three species. This is work I'm doing with funding from FAPESP Brazil and MinCiencias Colombia with South American collaborators in Campinas and in the Universidad Nacional de Colombia. So structure is, divergence is not big between north and south. And in the tierra firme species, you do have more outliers for FST. And they consistently group, so the southern groups consistently group. These are snapper calibrated trees using SNPs and using the coalescent and generation times to estimate ages. And what we find is that divergence between north and south, so here between north and south, is a couple hundred thousand years at most. So it really doesn't seem to have anything to do with the formation of this arc, but more so with how rivers have evolved later after the arc formed. So more with river incision dynamics starting at 130,000 years and not with uplift per se. And there's this wonderful paper if you're interested in how rivers form, especially about this area from Breda and collaborators explaining the process of how rivers form after they how they have formed in this area. Now we're running demographic models to understand how much gene flow there has been, if it has been in different time windows, etc. So I had worked with campesino communities in the Andes, but sampling in all of these rivers of Amazonia led me to work with Amazonian people and become really in awe of how much they know about biodiversity. And we wanted to show that. We wanted to show how important they are, how much they know, and how much they protect. So we moved. I had some experience with campesino Andean communities, but this is a whole different story. Cultural diversity in Amazonia is a lot bigger, linguistic diversity as well, and it's kind of a more remote area. In northwestern Amazonia, at least, it's a lot more remote, hard to work in. So we started by just working with occurrence data. So we produced these distribution models for all 260 Colombian palms. So Colombia has 10% of the species diversity for the world in terms of palms, and 260 species, and we stacked those distribution models, and we wanted to see if there are richness centers. And so in pink, you see the most species-rich places. And here in red, these are the polygons for collectively owned lands, indigenous collectively owned lands. So there's always been this debate, at least in Colombia and several South American countries, if collectively owned lands are good for conservation or not, if collectively owned lands are protected or not, or if they fall under the tragedy of the commons. And we wanted to show that there's high diversity in areas where there's collectively owned territories, but they're also away from threat. So where we find most threatened species, this is the map of threatened species, is really away from these collectively owned territories. So this just highlights the importance for conservation of collectively owned lands and cultural knowledge related to these territories. So we teamed up with leaders from different communities, from the Bora, Muri Muinane, Ticuna communities to work on Astrocarium because we had been sampling it, because we were more familiar with that system, and with graduate students, Lina from Colombia and Carmen from ASU. And we came up with this research question. So this is Olmar's home. These are Olmar's relatives, and this is Astrocarium palms. Are communities only accessing, so just like harvesting palms, or are they contributing to safeguarding and modifying biodiversity, at least in terms of these palms that are recognized and used by them? Is the genomic architecture and selection of managed versus wild species different? And we're starting now with Astrocarium because there are so many different species that are used and managed, but we have to start somewhere. So we're using Astrocarium because it's a system we know more about. And is their historical demography different, reflecting cultural uses? So can we understand, by studying their historical demography, can we understand something about use, the history of use of these species? And this ties to two important models in anthropology and archaeology, which is like a paradigm shift in South America, and in particular in these communities from northwestern South America, which is the well-known shift between hunter-gathering and agriculture, because uses of plants are so different, and the way you use landscape and you use land is so different in the two different paradigms. So hunter-gatherers are generalists. They use many, many species. They only collect parts of native plants. They're highly mobile. They move around a lot. They don't affect plant densities as much. They exert low selective pressures, and they work with mostly wild genomes. Whereas in agricultural societies, people are more specialized. They replace the standing vegetation. They're more sedentary, so less mobile. The plant densities are determined, chosen. There's high selective pressure of the used species, and genomes are more managed. But the Amazonian system is somewhere in between, because many wild species are grown, but not within traditional agriculture, right? They're grown or enriched in nearby forests, and they use some species that are not native from that site, but many of the native species. So it's what has been termed the hunter-gatherer horticulturalists. They create these gardens that they tend, but they also grow things within the forest. So it's like managed forests. It's a different system. And it's connected to what's called the Chagra systems. So they are still generalists. They use many, many, many species, including many palms. They grow and collect parts of native plants, but they don't entirely replace what's standing. So they keep some of the native species. They increase the density of some of them, so they will increase the densities of their used species. They're highly mobile, so these homes are moved around and they are highly mobile, but we don't know about their selection pressures, how managed the genomes are, and if they are structural effects on the genomes of these species or not, if we can find some sign of, not domestication, but of management at least. And if they behave as wild species, and why are they seem to be adapted to these anthropogenic systems. So Astrocarium chambida is one of these species that you can rarely find in deep forest. So it's native, but if you get away from humans, you no longer find it. It's managed, used for fibers. So it's a source of fibers, and it's rarely found within deep forests, mostly around humans. And it's used this way. So it's used within these managed forests. It's harvested. The young leaves from young individuals are harvested. The fibers are extracted, washed and dried, then dyed, and then woven into bags, hammocks. And its sister species, so this is chambida, the managed species for fibers, and its sister species is sympatric, not managed, not used. So we saw an excellent opportunity to compare if there's something different about their genomes, because they're sister, they're sympatric, but one is managed and the other one is not. However, we haven't been able to flash freeze. And you know this is an essential part of getting good genome assemblies, because you need long reads, and it has been difficult to flash freeze and export because you need all these permits and it's a long process. So can we do it without flash freezing? Yes, we can, but we need a lot of help. So we've been working with these different institutes. And what we're basically doing is we're extracting high molecular weight DNA from tissues fixed in ethanol. Then we're doing chromatin, the high C sequencing from tissues fixed in PG Shield, which is this solution only under testing. And then we're doing RNA sequencing from tissues fixed in RNA later. So when we go to the field and we collect in all of these solutions plus the herbarium sample. And from silica dry tissues, we also try to get PacBio long reads, and we do get something from them. However, what we got is like 1/8 of the expected yield. And when we assembled, it was like 2/3 of the genome. So these genomes are rather large, 3 to 4 gigabase pairs, and we got 2/3 of the whole length of the genome. So it's satisfying. You get something, but you won't get chromosome level, fully resolved whole genomes if you're working with silica dry tissue. And you won't be able to annotate them because we won't have the RNA. So this is where we're at with this project. We don't still have results, but I still wanted to show you because this is where we're going. Running late a little bit. Okay. So yeah, in the future, I still want to continue working with palms around humans. I think they can tell us so much about anthropogenic systems, but also about what humans create. So humans create drier systems or systems that tend to dry more open environments. In particular, this species does very well in transitional systems. And I think they can tell us a lot about the evolution of people and of more disturbed environments. So we're teaming up with Montgomery Botanical Center and colleagues from ASU to work on the historical demography. We are also trying to find, but we haven't been successful so far, but we are extracting more and more environmental eDNA from archaeological soils to see if we find this palm on archaeological soils. And to continue with my future goals, I've been very collections-based. I see the enormous potential of collections. In Colombia, I formed this tissue collection, which now covers all of the Colombian palms and has now 5,300 specimens. And with this tissue collection, I could, along with herbarium, work in herbaria. I could write monographs, describe species, work on red listing. So I think they're very valuable assets because they help prepare society, children, students, and researchers to become interested in the future of biodiversity. They're really time and space observatories facing biodiversity crises. They can guide our exploration strategies, so where, when, how to collect, right? And they're cultural repositories too, about plant and human species and species interactions. So I see the enormous potential of collections. And well, you have a great collection here, palm collection, the continent's largest palm herbarium. I've told several people that I've met that I started my journey with palms by working on some material that had been sent by Dr. Natalie Ewell to the person who was my supervisor during my PhD. So Cornell has always been a reference collection, and it is a reference collection for all palm researchers in the world. So with that, I want to leave time for questions, and thank you all for your attention. Thank you very much.
We have time for a couple of questions.
Yes.
First of all, thanks for giving us so much to think about. Marvelous presentation. Of course, I'm gonna ask you about the beetles.
Yes.
Yeah, and so not only the beautiful montane system that you showed us, but also the peach palms. It makes me think a lot of figs because those trees are feeding so much biomass, right? They're connecting the forest through frugivores, et cetera. They're really important. And all the focus on pollinator health in this continent and in Europe is very bee-centric. People are thinking about bee management, bee viruses, all of that. These beetles that pollinate your palms are never regarded as important, you know, in terms of their ecosystem services. So my question for you is, unlike figs, you know, they don't live their entire lives inside the palm tree. Where do these beetles live when they're not pollinating your palms? And is that part of the conservation question as well, to preserve their habitat?
Yeah, so this is why we really liked our results, because it shows the importance of congenerics, because we found in several genera, Astrocarium included, which I didn't show results of. But for Astrocarium, when you have different species of that genus living together, they keep the pollinator alive. They share the main pollinator and they keep it alive. So you do need assemblages to keep your main pollinator alive. It's not like you can go and plant a species away from its ecosystem and keep the pollinator alive because it takes a village to raise a child, because it takes the congenerics that share that same main pollinator. So that's the case for Astrocarium, for Euterpe, for Ceroxylon. We're finding more and more that's a pattern. For bacteries, it's a different group. It's Baridinae, another tribe. That's the so another tribe, but it's still beetles. And that's right. For palms, it's mostly beetles, and it's mostly unknown beetles, undescribed beetles, and miniature beetles. So they have just been disregarded, and I think they're an asset of course, of forests that we should look more into. So I think the way they keep them alive is there's specialization, but there's not. It's not. These pollinators are not exclusive to one species, so that they can share it around across the year. So there has to be some standing diversity to keep them alive.
That's another reason why monocultures don't work.
Yes.
Thank you.
And it's another reason why these communities that have lived for thousands of years in the Amazon know that the best way to get food is by growing or enriching their palms of interest within biodiverse systems, because they'll get a higher yield, and not by just extracting the one species and multiplying it by the hundreds and thousands, but by keeping it in a managed by biodiverse system. Mm-hmm.
Here in the Northeast,
Yes.
when the settlers came, it's estimated roughly 90% of our land was cleared for farming purposes. Surprisingly now, sorry, it seems to have recovered for the most part in terms of diversity. And currently, when we see some of these successional lands, they're a monoculture of goldenrod, but apparently that plays an important role in that succession. Can you speak to palm's role in natural succession back to forest from disturbed lands?
Yeah, so what you normally see for these canopy palms, it's different from the understory herbs. But what you can see with the canopy palms is when you have a high abundance of canopy palms, that usually coincides with an early successional stage. Why? Because they're monocots, they can grow faster than other trees, right? So they form these early forest. And then as the forest becomes more and more mature and the canopy becomes more closed and there's more shadow, then palms become less abundant. So you will still have the same regional pool of species, albeit in lower abundances, relative abundances for each. So palms do play an important role in producing biomass in those first early stages of succession. And then as the forest becomes more mature, they become less and less abundant, but still remain in the regional pool.
They're the goldenrod.
Okay, yes.
Yes. And they're very opportunistic because they love light. So an open corner in the forest is always an opportunity to grow faster and reproduce faster. So they use it.
You take one more,
Alejandra?
So you mentioned the volcanism during the Miocene, but you also have the incursion of the Sea.
Yes.
So do you think it have a role in the diversification or the increase in highly diverse populations that you have there?
Yeah, so the PEBA system has been more studied in terms of divergence because it meant a barrier for low elevation plants. Whereas this volcanism was like, we're studying it as a point of connection between mountain segments. So they're within the same region, but the PEBA system is a little bit northeast of where we're at with these volcanic centers. And these volcanic centers are on top of existing low topography. So they're acting upon mid-elevation lands, so already within the area of influence of the Andes, whereas the PEBA system was east of the Andes. And so they're similar in the way that both certainly affect the distribution of diversity. They're both certainly interesting because we can date them from different perspectives and see how the system comes together. And I know the PEBA system is specifically interesting because it left behind all of these species adapted to marshes and seasonally flooded areas. So like Mauritia and Manicaria and all of these species that were left behind by the Pebas system. So the Pebas system was a marine incursion, and it left behind all these swamps. And then the marine incursion retracted and further north, but it left all of these swamp adapted species. So it's funny because you're walking on tierra firme in the Amazon, and you're finding all these species that look like they're coastal. But it's because of, it's another example of how there are signs of the past everywhere if you know how to look for them.
So with that, I think we're gonna have to end the questions. If you have any other things, questions you'd like to ask, come on up. And don't forget, there's a lunch with the graduate students immediately now.
Thank you, everyone.
This has been a production of Cornell University, on the web at cornell.edu.